Okay. The mechanism is the most important part. Let's do the mechanism of you know, I have some of my best friends in Michigan from Dearborn, and every time I look at you, I think of them. I really respect those kids.
His father's brother, one of them.
Yeah, there you go. You know
He
them.
lives right down the street.
Say my hi. Say salam to them next time when you go. They're amazing people. Okay, now let's go over the mechanism. Okay, mechanism of. Let's do necrosis first. Okay, or cell injury, ischemia. See, ischemia can lead to necrosis or apoptosis. It depends what happens.
Are you redoing the process right now?
I'm doing the mechanism now. Yeah. So let's say ischemia. The important thing here is ischemia. Again, ischemia, hypoxia, I'll put both of them here. Causes very similar problems. Because even in ischemia, it's hypoxia that's an issue. Nutrients, it will take time, but lack of oxygen immediately affects you, right? So that's why. But which one is more severe, you think? Ischemia or hypoxia? Which one is more rapid? Let's see. Let's see. Yeah. You're right. You're right. Okay, so let's say no, there's ischemia and hypoxia. What happens? What's the big deal? Why?
Lack of blood supply and oxygen.
Okay, so I say hypoxia on top anyways. Hypoxia, lack of oxygen, so what's the big deal? Give me the mechanism.
No energy, no ATP. Mitochondria
Good.
not stable.
So this leads to lack of ATP. Lack of ATP. We have established this many times, that production of 100% energy requires oxygen. It depends on oxygen. Charging your phone depends on plugging it into electricity. Your electricity is mitochondria. And the source is oxygen, in a sense. Don't misquote me, I'm just giving you a brief overview in that sense. There's more detail to it, but without oxygen, you cannot make ATP. Period. So when it comes to ATP synthesis, there's two pathways. Oxidative phosphorylation and subcellular phosphorylation. Or anaerobic. You can say anaerobic glycolysis or aerobic. That's what I'm talking about. And you will spend a whole day on glycolysis, so don't worry. But I'm sure you were exposed to sometime in your life. Okay, so when you have less ATP, that means, and the reason of this less ATP is obviously no oxygen. Okay, so how would it affect things? What will happen to sodium, potassium, pump? So its functionality activity is decreased. Excellent, excellent. That's one major thing. Okay. What will happen to calcium pump? No, calcium pump activity. Decrease. Anytime you hear pump, what are you going to think? There you go, my homies. That's cool. Okay. All right. Now, excuse me. If there's no sodium potassium pump, what happens to sodium concentration outside or inside the cell? So sodium will influx. In other ways, sodium concentration inside the cell should increase. If you can't pump it out, that means it will increase, it will accumulate, right? You know that by definition there's 140 times more concentration sodium outside than inside. So if the channels are open, automatically it will come in. Now you can't pump out. And what will happen if sodium increases inside? What will happen to chloride? Aren't sodium and chloride cousins or couple? They're married? Yeah, wherever the husband goes, I mean, wherever the wife goes, the husband follows. I don't know. Make it other way around.
Real quick.
Yeah. Okay. And what follows them is children.
Water.
Water. That's natural. If you don't believe me, just take a spoon of salt. You're going to scream for water. You're going to have to take water because you'll feel thirsty. Okay. Now, so when this happens, that means the cell will swell. So some swelling will occur. True or no?
Yeah, true.
Yeah. Yep. That's common sense. So swelling, when that does occur, I'm running out of room. So 3 swellings are not good. One is mitochondria. Membrane will expand, it will be stretched if it swells, right? If the cell swells, the cell membrane will swell and will be stretched. If mitochondria swells, same thing. Although mitochondria is not done by this, I'll leave that for calcium. Let me just do this. Calcium, hold on. Calcium. Let's leave mitochondria for that. But lysosomes will swell, and what will happen to cell? And cell will swell. So lysosome and cells, these two will swell. Okay. And let's include one more thing. Endoplasmic reticulum. Let's say rough endoplasmic reticulum will swell as well. So subcellular structure will swell. Okay. Let's move on with calcium. What is calcium? The calcium is crazy, son of a gun. It just causes a lot of damage. You see, when it comes to calcium, a lot of enzyme activities depends on calcium. Enzyme activation depends on calcium. Enzyme activation depends on calcium. Enzyme activation depends on calcium. Okay. So calcium. So when you have the calcium pump not work, that means what will happen to cytoplasmic or cytosolic calcium? Increase. The pump is supposed to remove calcium from cytosol into endoplasmic reticulum and store it against a concentration gradient. That's why it's a pump. If you cannot pump calcium into the endoplasmic reticulum, obviously it's going to leak and accumulate where? Yeah. And rough endoplasmic reticulum. So let's just include here. Smooth endoplasmic reticulum also swells. When smooth endoplasmic reticulum swells, what happens to its membrane? It becomes leaky. It stretches. You know, if you have a clot or something and you have water, my grandma what they used to do is they would have these clots and they will put like liquid yogurt and the water will drip and it will become solidified cheese-like stuff. And if you stretch it, you know, more things leak out because the holes in the fabric will increase, the size of these holes. Same thing with membrane. If you stretch it, if it swells, you should expect leakage, increased permeability. Common sense. So if cytoplasmic calcium increases, this is the problem. This will increase activation of crazy enzymes. Remember that calcium is required for activation of intracellular enzyme. And those are, for example, lipase, protease. This is recording and it's, oh, that's good. It's fast too. Well, so lipase, protease, anything else? Nuclease, and let's talk about DNAs, RNAs, and many other enzymes. So it's good enough for now. Amylase as well. Phospholipase. This is important. Actually, let's include one. Phospho. I'm missing an S. You can add that. Phospholipase. Now, phospholipase will damage what? Membranes. Excellent. Membrane. RNAs and DNAs? DNA and RNA. Nucleus. And the chromosome structure is defective. Now, if you don't have the genes, can you repair the cell? No. Okay, so protease, proteins, lipase, other lipids. Okay. So again, this is cellular damage so far, just simply because of what? Lack of ATP. Okay. Now, if smooth endoplasmic reticulum is damaged, it will lead to increase in calcium. Right? It will lead to increase in calcium. If lysosomes are damaged, What will happen to lysosomal membrane when they swell? Burst. And that's irreversible. When lysosomes get damaged, the injury becomes irreversible. Because what is lysosome filled with? Digestive enzyme and acidity, and that will digest the heck out of that cell. Okay. Now let's do one more thing. How about if the cell itself swells and the membrane becomes leaky? Yeah. It gets worse and worse. So the irreversible injury is usually membrane damage. Membrane damage. Especially lysosome membrane, mitochondrial membrane, and cell membrane. And we haven't talked about mitochondrial membrane yet. Okay, so now we come to the mitochondrial membrane. Mitochondria is made of two membranes, right? Mitochondria is made of two membranes. So let's say on the inner membrane of mitochondria you have these enzymes, which are known as electron transport enzyme. They transport electrons. All of you heard of it. In this case, let's make this enzyme. Let's call it cytochrome C, or cytochrome C oxidase. Cytochrome C oxidase. This oxidase requires what? Oxygen, yeah. The whole idea is oxygen. Oxidase depends on oxygen. Okay. And then on the inner membrane you also have transport channels. Okay, so proton channels. Let's just put it this way. Proton channels. Protons are hydrogen ions. And these proton channels, they become very leaky. Okay, so proton leak out. Two things. Cytochrome C oxidase is a signal for apoptosis. This is a signal for apoptosis, so it causes apoptosis. Whenever you find cytochrome C oxidase in the cytoplasm, it means apoptosis is going to initiate. Okay, and if the mitochondria loses its protons, it's the proton that drives the pump to make ATP. ATP synthesis depends on protons. So let's say concentration of hydrogen ion within the mitochondria decreases, or in the cytoplasm it increases. In the cytoplasm it will increase. Okay. So concentration of hydrogen ion, let's just put here cytosol. And the cytoplasm increases. When this increases, what happens to the pH of the cytoplasm? Drop. And when you lose the protons from mitochondria, what happens to mitochondria? It's non-functional. So mitochondria will become non-functional, and that will lead to irreversible injury. Okay? So that's one thing. When mitochondria is not working, okay, because there's no oxygen, that means your cell will start using what process to make ATP? Anaerobic glycolysis. The end product of anaerobic glycolysis is lactate or lactic acid. So lactic acid will increase, and that means decrease in pH, and that will further damage the cell. Okay, so this is what would happen in terms of ischemia. The initial step is lack of ATP. Hypoxia and ischemia. The initial step is lack of ATP. And it depends which side we go to now. But irreversibility depends on mitochondrial damage, cell membrane damage, and lysosomal damage. All three membranes. But if I use the term membrane damage, that's good enough. Unless I say specifically, the most important membrane is mitochondria. Because that's the source of ATP, right? Okay, so that's ischemia. Now, ischemia can cause necrosis or apoptosis. If the cell bursts If it's cell membrane damage, it burst. Now let me just do this. Now let's compare apoptosis and necrosis. You know that at the end of the day, ischemia or hypoxia leads to multiple things, multicellular damage. The most important thing is mitochondrial membrane damage, cell membrane damage, and lysosome. When it's done, the cell is irreversible. Okay, only then apoptosis or necrosis can occur. But let's see, we have membrane damage. So ischemia will lead to decrease in ATP. Let's keep that. And then let's say there's two things that could happen. Let's make this mitochondrial damage. Mitochondrial membrane integrity is disturbed or damaged. And here you will have on this side cell membrane damage. It depends which one comes first, right? Depends which one comes first. Remember, the unique thing about apoptosis versus necrosis was in apoptosis the cell is intact. The cell does not burst. Cell lysis doesn't occur. In necrosis, cell lysis occur. So if cell membrane is damaged, what would that lead to? Cell lysis, right? So this will lead to cell lysis. If mitochondrial membrane is damaged, this will lead to release of what? Cytochrome oxidase or cytochrome C oxidase. If cell lysis occur, that means cellular particles are now swimming in extracellular fluid or blood. If that's the case, that means they will recruit what? The army, the police, the this, the that, or inflammation. So this will lead to inflammation. And this will lead to necrosis. Okay? In necrosis, the first cells that come to the site of inflammation are neutrophil. Signs of acute inflammation are what? Neutrophils. So the major cell type in coagulative necrosis is what? Neutrophils. A week later, there will be macrophages, not immediately. A week later. The first few days, it's neutrophil that will start engulfing this dead tissue, swallowing them, this dead tissue to clear up. It's almost like garbage trucks. They will come and clear up the area. That's what neutrophils will do. Remove the damaged cells. Now, if cytochrome C oxidase is released, and this is a sign for what? Apoptosis. Now, apoptosis has few steps, but at the end of the day, the major enzymes, the major thing that identifies apoptosis, the enzymes are known as what? Caspase. Caspase. Wow. I try to choose good colors, but then it becomes, you can't see anything. For whatever reason, the last few weeks, I always use green, and some students tell me, like, my eyes are hurting. It's too bright. At least the ASMLE students said that. Caspase. Now, caspase stands for, okay, caspase stands for, the aspase stands for aspartate. Aspartate, or aspartic acid. Okay, that's what it stands for. These enzymes, caspase, are the enzymes of apoptosis only. They only cut at aspartate location, or if a protein is made of aspartate, they only cut there. They only cut at aspartate residues. And the C stands for cysteine. C stands for cysteine. Okay? Cysteine protease. What is a protease? Good. So caspase, they have two things. Number one, their active sites, their mouth with which they break things, they have cysteine. They're cysteine protease. That means they have this property of cysteine protease. But they cut where? At aspartate location. That's where it comes from, caspase. So their teeth are made of cysteine. Their teeth are made of cysteine. That's how they break things. But where do they break things at? Aspartate location. A protein that has residues, aspartate, that's where they cut. Again, when you learn about protein, you will know that. But at the end of the day, that's what caspase do. There are about 10 enzymes, caspase. So, number one, by mitochondria, the one that's active is caspase 8. And this is called the initiator. It's called the initiator. And eventually this will activate caspase 6, I think, or 3. And then these are called the executors. They actually do the damage. They'll start killing the cell. Okay? So initiator is 8, executor is number 6. Okay? Alright, so that's the difference. At the end of the day, what caspase is, they also signals macrophages to do phagocytosis. Macrophage. So again, apoptotic cells, the end of apoptosis, phagocytosis done by macrophages, end of necrosis primarily by who? Neutrophils. Primarily. So that's one more difference. Macrophages will come and eat those cells that have been damaged. Yeah.
What determines which way is going to go when decreasing ATP?
Depends if mitochondria gets damaged first or cell membrane.
Well, both of them they have a lot of ATP, so which one is going to go first?
It depends on the circumstances. As I said, it depends if the mitochondria gets damaged first or the cell membrane. Both membranes. Depends which membranes.
Is there any specific?
They're the same membrane, so it depends on the... If the internal environment is severe, that the mitochondria gets damaged first. And some cells have the cell membrane is much stronger. For other cells, the mitochondrial membrane is stronger. So it's a random thing. Okay. All right. Let's go to apoptosis now. Apoptosis. So in apoptosis, again, the cause of apoptosis is usually DNA damage. When would you want to kill your own cell?
Quick question, sorry. Like, I think I confused myself. Ischemia will lead to mitochondrial membrane damage, which then leads to apoptosis?
Mm-hmm.
So it's not...
So hypoxia or ischemia can either cause apoptosis or necrosis. That's what Mazen was asking me
So
too.
mitochondria is apoptosis.
Cytochrome C oxidase is a signal for apoptosis.
But initially it's ischemia leading to necrosis of mitochondrial membrane.
Let me just do it again. Good. So let's do this, guys. Look, ischemia occurs, and you know that ischemia and hypoxia leads to decreased ATP. True? Okay. If this leads to mitochondrial damage first, then apoptosis will occur. If this happens to be causing cell membrane damage, then necrosis will occur first. But you can have a group of cells that go through apoptosis, group of cells that go through necrosis. There's no such thing in your body like clear-cut this, that. Okay, both processes can occur at the same time. But depending on the individual cell, one cell cannot go through both things at the same time. A single cell cannot go through both things at the same time. If the mitochondria gets damaged first, then that means cytochrome C oxidase is released first. This signals for apoptosis. If cell membrane is lysed, if the cell is lysed, then there's no room for mitochondrial damage. The cell is just dead. So it depends if the cells burst first or mitochondria burst first. If mitochondria lyses first, then it will lead to apoptosis. If the cell membrane damages and the cell goes through lysis first, then it's necrosis. Does it make sense?
So cell membranes first, it's necrosis for mitochondria.
Yeah.
I guess the confusion really came from associating ischemia with necrosis.
Yeah.
Ischemia can cause necrosis or apoptosis. Apoptosis has some specific causes, but
So the whole
Yeah.
time is associating ischemia with necrosis.
yeah. Okay. All right. Okay, good. Yeah, overall, by far, ischemia is number one cause of necrosis, by far. But there are circumstances where ischemia can even lead to what? Apoptosis. Okay. For example, if the cell is damaged to the point of no return, your body's not going to tolerate that. You say, you know what? I have the ability to replace it with new ones. So why would I want to keep a damaged cell? You know, so again, when we have in our communities members that are toxic to the rest of us, what do? We put them in jail and things like that. Same thing with your body. When there's a group of cells that don't benefit you but can cause harm, let's say they're cancer cells. What do you want to do with cancer cells? They can turn other cells into cancerous. So what are you going to do? Put them in jail or inject them with something like apoptosis. So as long as you can use common sense, you'll be okay. Okay? But guidance is required sometimes, I have to remind you, and it helps. Again, apoptosis it's a defense mechanism. You don't want damaged cells to persist. You don't want mutated cells to live forever. You don't want these diseased cells because they can cause further disease. For the safety of the entire body, you have to sometimes have your cells commit suicide. Okay? So your cells understand that. Your cells have this ability to understand. For the well-being of the functionality of the body, some cells have to commit suicide or have to be killed. I mean, it's a harsh term. Maybe my terminology is not accurate, but I think it gets across. So I could be polite or I could be harsh, but that's the idea. For the survival of you, sometimes your body have to kill its own cells, and that's apoptosis. Sometimes the cells themselves will say, hey, you know, get rid of me because I've been infected. I've been damaged. I'm no longer in my control. I can cause more damage. This condition can spread. So your body has to go through the process of apoptosis. Now, necrosis is not apoptosis. Necrosis is actually external damage. Okay? It's not for the benefit, it's actually destructive. Apoptosis can be destructive or beneficial. Necrosis is never beneficial. Never beneficial. In a sense, like, let's say, necrosis will be, you know, like a murderer committing murders, serial killers. But taking that serial killer to kill them by your own police for the benefit of others is apoptosis. Okay? All right. Generally, cells want to survive. The idea is, you know, like, why do you have children? So you can exist over and over again. That's the idea. You might not recognize that, but that's the idea naturally. Because human beings wants to live forever. Wants to live forever. Wants to live forever. Cells wants to live forever. Cells. You are a cell. Cells, naturally, they have inclination in them. They want to survive. They don't want to die. They don't want to die. That's why they scream every time when you hurt them. And you say, oh, I hurt, I have pain. Because they're telling you, I don't want to die. Don't cause damage to me. Don't cause harm to me. So these are the mechanisms as far as science has discovered. Okay, they're limited in terms of understanding, but it's something, right? It's something. Because based on this, we can actually treat conditions and diseases. All right, let's move on. Let's go to apoptosis. Now, apoptosis, again, it could be caused by DNA damage or mitochondrial damage. Or lack of growth factors, growth hormone and things like that. Okay? Growth factors, again, you can put under growth factors multiple things. You can put their thyroid hormone. You can put all hormones if you want, all interleukins. Those of you who don't know about growth factors, anything that causes growth. We have hormones that are responsible for growth. For example, without LH and FSH, what happens to your ovaries? For girls, no guys, because I'll ask you separately. Girls, when you have, no guys, only girls. Questions only for girls. When you don't have LH and FSH, luteinizing hormone and follicular stimulating hormone produced by your ovary, right? I mean by your anterior pituitary, acts on ovaries. If you cannot produce that, what happens to your ovaries? They shrink. They go through atrophy, and that is apoptosis. So lack of growth hormone stimulation, lack of growth hormone stimulation, growth factor stimulation, cells will go through apoptosis. That's one signal. Interleukins. They're usually abbreviated IL. They're also growth factors. Now, the major growth hormones, growth factors in terms of hormones are insulin-like growth factor, insulin, growth hormone, and so forth, which is a discussion in endocrinology, right? We'll get there. Okay. Let's see if this makes sense. Look, if your mitochondria is damaged, do you think that cell can survive? No. If you don't have the machinery, the machinery, let's say, in this case, I don't know what's a good example, but anyways, even in wars, when people go to other countries and do stuff, they destroy what? The source of survival. Infrastructure. The hospitals, the schools, and the factories, and all those things. For what purpose? Because they want to starve you. They cannot get you. They want to starve you to death. So if mitochondria is damaged, you can have all the oxygen available. What's the purpose? Because mitochondria is what consumes oxygen to make ATP. The source of your energy is what? Mitochondria. When the cell recognizes that the mitochondria is damaged permanently, it's common sense for the cells to go through apoptosis. Why would you want to keep it? You don't have to wait for a virus to come. You don't have to wait for ischemia to come. You don't have to wait for anything else. So no necrosis is needed. It's a programmed cell death now. Okay, so mitochondrial damage makes sense if it's damaged. The source of survival, it's the source of ATP for a cell. If the mitochondria is damaged, then that means the cell cannot return to health. It's irreversible injury. Why would you want to keep an unhealthy cell? I know it sounds cruel, because if you translate it to humans, because we have people on feeding tubes and stuff like that and blah, 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 right? A lot of ethical issues occur. But yeah, some people will come and say that, we believe in God, the cell can recover. Yes, it can. But the recovery is not as common as destruction of it and causing more damage. So a lot of things can happen, of course, but when you have knowledge, you have to make a decision. Sometimes decision can be cruel. So the cell, your body at that point makes a cruel decision. Like even that cell you can call the handicapped, special cells, whatever names you give them in terms of society's understanding, but that cell is damaged and it's not viable for your health, so it will cause more damage to your body. So what do? Your body says, let's peacefully kill it. Okay? So that's mitochondrial damage. Now, when it comes to DNA damage, what's the point of DNA? Let me give you an example. If one human being turns out to be alien, after being a human being, he switches, mutates to alien. And the job of that alien is to start killing human beings. What would the rest of the people do? Kill him. Unfortunately, right? That's what it is. Killing is a strong term, but that's what it is. So because that person, if he has more children, they'll be like him. And eventually, if that population persists and stay and grow, humanity will go extinct. Same thing with DNA damage. DNA damage is a problem because if you have mutation in DNA, that can lead to further mutation, and you'll pass on to children upon children upon children. That means you're decreasing cell survival. Again, not all mutation, those mutations that are damaging to cell. So therefore, in this case, usually cell cycle. When it comes to DNA damage, let's say this is a cell cycle. Cell cycle has many stages, G1, S, and G2, which we'll talk about on Wednesday. There's a checkpoint and there's a checkpoint. Here is S stands for DNA synthesis. So DNA is synthesized in this location. If there's a mistake in DNA synthesis, you're not going to pass that on to children. So what do? You stop this right here or right here. And P53 is a gene or a protein that is a checkpoint. It's a tumor suppressor gene. What does tumor means? Increased growth. What does suppression means? Preventing. So this P53 is tumor suppressor. It prevents tumor growth by what? By stopping the cell cycle when they recognize there's a mutation. When P53, it's just somebody like a security guard sitting there. Oh, there's a mistake in DNA. You cannot continue. And it puts a stop to that replication of that cell cycle. So DNA damage is a serious thing. Because DNA damage can be permanent. When it's permanent, you'll pass on to children upon children upon children. And that can lead to more abnormal cells. And you don't want abnormal cells because it will make life of normal cells very difficult. And it will cause disease, and you'll be hurting, you'll have pain, you'll have to go to doctors every day, and so forth. So hopefully all of this makes sense. Now, growth hormone, or growth factors, they're required for cellular growth. If they're not there, the cell will not grow. They will atrophy. And when they atrophy, they have less function. If they have no function, they're just useless then. And then they have to go through apoptosis. You know, cells have specific function. If they cannot perform that function, what's the point of having a cell? What's the point of having a cell who is designed for something specific and they're not doing that? A lot of people translate that philosophers into human being. Human beings have specific functions to perform. And if they don't do what's their purpose, it's useless. And they will not survive for long. You know, so because people believe in death and life after death. Those who don't perform their purpose in life, they are unlikely to survive after death. Meaning that. But anyhow, actually I think like this, so I just blame others for it. So let's start with this. Okay. So when DNA damage, this DNA damage activates, for example, caspase. Before caspases, there are sensor molecules, okay? And mitochondrial DNA damage, these are sensor molecules. Let's say, for example, I think I remember the mitochondrial one. So the sensor molecules are known as BID, BAD, and I think BIM, I think so. We'll just include these two only. Okay, so sorry, just keep this one. I don't remember the other ones. There's 3 more. And these guys will activate first. So these are sensor. What do they sense? Mitochondrial damage. Oh yeah, sorry. When mitochondria gets damaged, what do they release? Cytochrome oxidase. And this will activate. This is sensed by who? By sensor molecule. And these sensor, one of them is BID. And then these guys will activate pro-apoptotic genes. So these are sensors. This senses what? Permanent damage. Okay. And then you will have pro-apoptotic genes. In this case, one is known as I think DAX and BAX, something like that. BAK, right? And then DAX? BAX, yeah, BAX. There you go. These are the one I was looking for. These are pro-apoptotic genes. Pro-apoptotic genes. And these guys will further activate caspase. Okay, they will activate caspase. Okay, they'll activate caspases. Same idea with DNA damage and hormone and growth factors. Now, there's one thing I want to know. So let's say this is the mitochondrial membrane. On this membrane, you have pores through which larger things can actually diffuse out or in. These pores are closed by, let's say this is closed. You can call it on the outside or inside. So these are closed, locked in. There you go. Okay. This molecule is known as BCL2. So BCL is anti-apoptotic. BCL is anti-apoptotic. It's not pro-apoptotic. Does it make sense? Let me make one more channel. Hopefully that'll make sense. I heard a lot of clicks. That's good. Of pens. When I hear that, I'm like, yes, people are learning. When I don't hear it, I get nervous. Okay. Red is BCL. You see BCL covers what? The channel on the mitochondrial membrane. So if that channel is closed, can cytochrome P, cytochrome oxidase, can it leave? No. So if it doesn't come to mitochondria, no caspase will be activated. So it's anti-apoptotic gene, right? BCL is anti-apoptotic. It's not pro. But guess what? Now these other molecules come, known as BAC and BACs. They're pro-apoptotic, and they come and they attach. When they attach, they will remove BCL out of the way. And now things can leak, cytochrome can leak out, and this will cause apoptosis. BCL stands for B-cell lymphoma. B-cell lymphoma. There's a huge family of these molecules in genes. They all have one thing in common. They cause what? They prevent apoptosis. So now, if you prevent apoptosis, sometimes apoptosis should occur. Some cells should die. For example, like white blood cells. After they have done their job, they should be replaced with new ones. They should be recycled. If a white blood cell is full of debris and old particles, you have to kill it and remove it, so because it won't be functional anymore anyways. So they have to go through what? Apoptosis. But if they don't go through apoptosis and they have overexpression of BCL2, which prevents apoptosis, what happens to white blood cells? They're immortal. They live forever. And what do you call that? Leukemia. Leukemia. So most leukemia will have BCL mutation or overexpression of BCL. The major leukemia, CML, ALL, and AML, for example, the major leukemias, they will have overexpression or overproduction of BCL2. They won't die because they have something that is protecting them from apoptosis. Okay? Okay, so I hope you understand. Pro-apoptotic genes and molecular what? BAC and BACs. Anti-apoptotic is what? BCL2.
So BCL2 is removed by those two. Can be removed by those two.
Okay. Now let's... Malcolm, you had a question. Remember a long time ago? Autoimmune diseases.
Yes, autoimmune.
Autoimmune diseases. Good. Now, the thing is, T cells, when they do their job, okay, when they do their job, they're done. And you have to get rid of them. They'll express on the surface Fas ligand. Cytotoxic T cells. T cytotoxic cells. Cytotoxic cells. C cytotoxic T cells will express on the surface Fas. Fas is a receptor, okay? Fas is a receptor. Fas ligand is a ligand for this receptor. Only cells that express FASL can be killed by apoptosis. This is a tag for apoptosis. Somebody asked me earlier, right? I said, wait till we go to mechanism. FASL, think of L, lethalized. Their tag, when you put a tag, you know what, let's do this. Sometimes pictures make life easier. Okay. There's the green cell. Mazen, guess what? I watched the movie last night. Do you know what that movie was? Something like this. Perfect Getaway or something? No? Okay, you guys never heard of it? I never heard of it too. But I just watched it by accident. I thought it was good. People who were a killer, they were presumed to be good, and those who were not killer, they were thought so. Misconception. Where is the thing? Here you go. Okay, so we have three cells. Let's say, the tag For apoptosis, this dark circle. This is fast ligand. This cell on the surface expresses fast ligand. Let's say 2 of them is a signal. And the other 2 don't express them. The only one that's tagged for killing is what? The green cell. So cytotoxic T cells will come and attach to the fast ligand, and then it will kill them through the process of apoptosis.
Fast ligand basically activates apoptosis.
Mm-hmm. The end result. The end result of it. Okay. The end result. The swallowing of it. Because remember, apoptosis initiates with apoptotic body formation, right? And then for that to be swallowed, you need fast and fast ligand. Usually cytotoxic T cells kill what? Viral infected cells. Now, let's say a virus causes, I'm glad you talked because now I remember what you asked. Now let's say this is a normal cell, this blue one, normal cell. Now it doesn't express anything on its surface that looks like a FAS or FAS-L. After infection, they start, or some of the viral DNA is incorporated to the human cell DNA. And these cells start expressing what? Fast ligands. Now, you're killing normal cells. That's autoimmune disease. So failure of apoptosis can lead to what? Autoimmune diseases. In some cases, cells, your own cells, when they express antigens, they're abnormal, or there are normal antigen and they're recognized by mistake by other cells, that leads to autoimmune disease. Let me give you an example. Let's say, in the thymus, so sometimes knowledge from previous areas will be helpful too. 4 more minutes, then we're done. Have you heard of thymus, guys? Thymus, where is it located? In the neck. In the neck, right? There's thyroid, and then you come about 4 fingers lower, just about the notch, right here is the thymus. Thymus. Why are T cells called T cells? Because of the thymus. You take the T from thymus, call them T cells. T cells get education in the thymus. So thymus is Avicenna Medical Academy. And you guys are T cells. So T cell education occurs where? In the thymus. Now T cells are taught in the thymus, do not attack your own cells. Any of those cells, any of the T cells that attack your own cells, they are deleted. By what process? Apoptosis. So let's do this. You can have T cells. Let's say 2 types of T cells. In shape and everything, they're the same, right? What is the color of T cells? Blue. And how about their nucleus? They're huge. Good job. They're lymphocytes. Small cytoplasm, right? So you have 2 T cells, type A and type B. A does not recognize your own antigen. So that's normal T cell. Normal. B is self-reactive T cell. Somehow it escaped the thymus, let's say. It was not deleted. Or later on in life, you did something and these cells turned against you. Okay? But at the end of the day, you can have cytotoxic T cell I'm talking about. Cytotoxic T cells that are self-reactive. They attack your own antigen. Even your antigens are normal. Your cells are normal. That means this T cell is defective. So these T cells can be tagged with Fas ligand. Now, the normal T cell will come and attack these guys to clear them, because these will attack your own cells. Let's say there is your liver cell. You heard of SLE, right? You heard of SLE? Lupus, you heard of lupus? In lupus, you can have T cells that attack your DNA. They shouldn't attack it. Now, in thyroiditis, like Graves or Hashimoto, you can have, especially Hashimoto disease, you can have T cells start attacking your own thyroid. Would you want that? No. Okay, so then the normal T cells will come in because they'll express on the surface fast ligand, they'll attack them and kill them. That's also apoptosis. Okay. Again, at the end of the day, the thymus will only allow normal T cells to come to circulation. Abnormal are deleted. The last thing I want to do is sometimes cellular injury can occur because of accumulation of foreign things and internal things. Abnormal accumulation. Okay, let's talk about those. What if, in some cases, a cell can start accumulating? Let's put it this way. Let's say your house is a cell, corresponding to a cell, and the garbage people are on strike. And then you keep accumulating garbage for 1 year. And you cannot throw it away outside. What will happen to your house? Eventually you get sick. Same thing with this. So when you have even normal things, when they even water, look, if water abnormally accumulates within a cell, what happens? The cell will burst. It will burst. So toxins, the term toxin is a relative term. A toxin can be therapeutic at some dosage. At higher dosage, it can be toxin. So a molecule can be good for you in an appropriate dose or concentration. If it's too much, it can become toxic. If it becomes too little, it's deficiency again. So everything in moderation or in balance in that sense. So lipid. Now lipid storage in cell is known as steatosis. Okay, or fatty change. So the term is also used, fatty change. Usually this happens in liver and muscles. Liver and muscles. Fatty change. The triangle stands for change. Steatosis. This usually happens where? In liver and skeletal muscle, cardiac muscle, especially cardiac muscle, and smooth muscle. So muscles and liver, or hepatocytes. Lipids can also, so this is actually accumulation of triglyceride. Sorry, I should have said that. Steatosis is abnormal accumulation of triglyceride in liver cells, muscle cells. Can happen in other cells as well, but usually in this. And when it does, so let's say this is a cell, normal cell, let's say it's blue. Now steatosis, what happens, it will fill up this cell, it will accumulate and accumulate. The nucleus will be pushed towards the side. The nucleus of that cell will be pushed towards the side. And then the cell will appear as slightly yellowish and clear, huge cytoplasm. They will look like ring cells. What's her name? That singer. Put a ring on it, whatever. Fiance? Okay. Fiances do put rings on people. Okay, so this is triglyceride storage, and it will be called steatosis or fatty change. Now, atheroma is abnormal accumulation of cholesterol, not triglyceride. Cholesterol. And it occurs where? Within blood vessels, especially arteries, blood vessels. Amongst blood vessels, arteries. Xanthoma, again, is accumulation of cholesterol. Abnormal accumulation, not normal. It usually occurs under your eyelid, on your ankles and tendons and stuff like that. On the face and eyelid, they look like yellow deposits. It doesn't look good. That's xanthoma, and it's because of abnormal cholesterol. Usually, people would have hypercholesterolemia, familial hypercholesterolemia. It's a disease we'll learn in lipid section. So look, we cannot live without lipids. You know that survival depends on fat and lipids, but they can accumulate genetically, environmentally, and sometimes different causes. So these are the names. Please remember these names. You can have something known as, let's say, Bilirubin, right? Very good, bilirubin. I'll do only 2 or 3, the rest you can do because it's all in your PowerPoint. Bilirubin accumulation. Bilirubin is a byproduct of hemoglobin, a breakdown product of hemoglobin. Bilirubin is insoluble, so it deposits in your body tissue. If it does, it will cause jaundice, and it'll give you yellow color. The skin in conjunctiva and sclera of your eye could turn yellow, and you can recognize that. That's abnormal accumulation. It's indication of a disease state. Now, steatosis, usually you cannot see them superficially. You have to do microscopic examination of cells, and the cells will appear as clear. Clear. Just in the corner, one area, you will have a dark blue staining nucleus. And the nuclei always stains blue. Why is that? Because they will have chromatin or chromosome, which stains blue. They are basic. What if you have abnormal accumulation of iron? Hemochromatosis, hemosiderosis. What if you have abnormal accumulation of melanin? Dark spots on your face. Let me see what else. What if you have abnormal accumulation of what else is there? How about smoke particles? That's known as pneumoconiosis in a sense. Coal particles. You can have silica from sand. You can work in a nuclear plants and battery plants, and you can have berylliosis. So anything abnormal from outside can accumulate in the cell and cause diseases. But the most common route is what? Inhalation, right? So go over this. It's in your PowerPoint. It's one sentence definition. That's about it. So as long as you know the names, and that's what's accumulated in the body. Oh, one more thing. Let me just do this one. I remember this is important. Hyaline change. Hyaline change. This is usually abnormal accumulation of protein or protein-like product. Protein or protein-like product. Okay. And it appears smooth, glossy, eosinophilic in color. What is the color eosinophilic color? Pinkish. So they'll appear in terms of appearance eosinophilic, glossy, shiny, and smooth. And it's homogeneous, uniform. There is no variety in shape or whatever. All looks like a shiny glass. Hyaline change. Okay. So look over these accumulation. That should be important. Okay. I think you will have 2 or 3 questions from accumulation. One on apoptosis, 2 or 1 on necrosis, and blah blah blah. I've done that. So if you have any questions, otherwise we're done. Yeah.
From the beginning, what is the difference? I just need this for my own clarification between anemia and hypoxia.
Okay.